Hello everyone. In this video, I'm going to present you about Salmonella. Classification of Salmonella. It belongs to kingdom Bacteria, phylum Protobacteria, class Gamma Protobacteria, order Enterobacteriales, family Enterobacteriaceae, genus Salmonella. History of Salmonella. Its history shows that this bacterium was first discovered by a scientist named Daniel Elmer Salmon in 1884. He isolated this bacterium from the intestinal tissue of pig. From the discovery up to the 1980, about 30,000 people were reported as infected in USA. The epidemiological study and the research was continued from the 20th century, and finally the CDC reported as a 1.4 million cases per annually in USA. Introduction of Salmonella. Morphologically, Salmonella, on the basis of staining, it is gram-negative. It is bacilli in shape, means having rod-like structure. Its size is about 1 to 3 or 0.5 microns, and they are motile, having peritrichous flagella structure, as you can see here in this picture. General characteristics of Salmonella. They belongs to Enterobacteriaceae family, means they have ability to live in the intestine, means these are the GIT bacteria. Beside this, in the absence of oxygen, when they live as an aerobically, so they don't have ability to ferment lactose and H2S positive. Along with that, their special habitat is intestinal tract of animals. But if in rare cases they enter in the intestinal tract of human, then they will cause pathogenicity over there. In this GIF, you can see here the peritrichous movement of Salmonella. Culture characteristics of Salmonella. They survive as aerobic and as facultative anaerobic. They grow on a simple media known as nutrient agar. They have ability to tolerate from 15 to 41 degrees centigrade, but as a mediumly they survive on 37 degrees centigrade. Their colonies are generally large, but their colonies looks like a low convex structure. On max concave agar, their streaking generally appear as colorless. They are also cultured on a selective media like Wilson, Blair, Bismuth, sulfuric medium. Antigen structure. Most of the peritrichous flagella containing having H antigen, and those which contain capsule they have K antigen. Means we can say that antigens are found not only on the pili, not only on the cell wall, but they are also sometimes found in the cell membrane as well. Pathology of Salmonella. How pathology is caused. Actually, these bacteria enters through the digestion processes and finally they attack to the microvilli and its tissues. Then they also attack to the mesenteric nodules found in the intestine. Then they enters in the bloodstream and cause different infections. Classification of Salmonella on the basis of serotype. Salmonella are divided into two main categories: typhoidal and non-typhoidal. The typhoidal are those which causes typhoid fever and paratyphoid fever. Their examples are Salmonella typhi and Salmonella paratyphi A, B, and C. The non-typhoidal causes gastroenteritis and food poisoning. They are the non-serotypes. In this graphy, we will know about the transmission of this bacterium. As you can see here, this is the fecal oral route transmission. Whenever a person is not curing and not washing hands, and his hands are contaminated when he is eating food without washing hand, so he may be get infected with these bacteria. So, for example, whenever he is eating a raw meat or eggs or poultry or using unpasteurized milk, so there may be the presence of these bacteria, so he may be infected with these bacteria. Or this can be transmitted also through the direct contact with animals like reptiles and the birds. Whenever a person is touching in a direct contact with reptiles or a bird and they are not washing hands properly, so there may be the transmission of this bacteria. Because as I already told you, okay, they are mainly found in the animals, so their habitat is animals, so that's why. Along with that, whenever in rare cases this bacteria is going to the intestine of a human, so they causes the pathogenicity. Like this bacteria is going to the intestine of a human, so it has invaded the microvilli and they are dividing continuously in the microvilli, so they are increasing in number time by time. And now you can see here they have produced their eggs, and along with that they have released some pro-inflammatory cytokines chemicals that has inflamed at that area. So due to these inflammatory responses, such signs and symptoms may be appeared, and like this case, the person will feel severe diarrhea and there will be abdominal pain in the intestine of that person. Means this is a sign and symptom of gastroenteritis, or he will feel food poisoning. It is a sign of a non-typhoidal bacteria. When these bacteria further multiply and deeply penetrate into the microvilli, they change to another stage known as bacteremia, and this stage will cause enteric fever. So, due to enteric fever, a person will be suffering from heart problem, lungs problem, bones problem, etc. Clinical syndrome of Salmonella. The diseases caused by Salmonella are generally called as a salmonellosis. There are the few words that I am going to explain here, like enteritis, that is also known as a acute gastroenteritis. Enteric fever, this is related to the typhoid fever. Septicemia, it is actually the infection caused by the bacteria. Moving further, the symptoms shown in the infected livestock animals, including, like here you will see that acute enteritis. This is its general symptoms are a fever and a severe diarrhea to the animal. Now you will see that this is a cattle, and you will see that how she will be in a situation of a diarrhea. Now, diagnosis. It is done on the basis of clinical signs and symptoms that can be easily confirmed by culture method. We have to culture of stool, blood, urine, or tissue, so final result can be achieved easily. These are the some sort of the samples through which we can easily diagnose uh, this salmonella, that what kind of a bacteria is or not. Biochemical tests. There are some biochemical tests mentioned in the picture, like TSI test, glucose test, oxidase test, indole test. Now, preventions. Prevention includes following precautionary measures like wash hand properly and sanitize them, proper handling of the raw meat, and cook the food properly, including meat properly. And you have to be cured from the contaminated animals and their products like their meat and eggs. So these are the some sort of the precautionary measures to which you have to follow, and you can protect not only to your animals but also to your health from these type of bacteria.